So now we're going to have our second team talk for today. In our first talk, we heard a lot about the functions of neurons, and this talk is going to focus more on what these fundamental units of neurons are. This is one of the big questions at the Institute in the cell types program, and there's a lot of different methods being used in order to characterize different cell types. And in this talk, you're going to hear about work being done using single-cell transcriptomics to determine the different cell types. The title of the talk is Transcriptomic Cell Types Across Cortical Areas and Species, and representing the team is Basilika Tasic, Trygve Bakken, Zizhen Yao, and Rebecca Hodge. Please join me in welcoming them.
So we will give you an overview of where we are today on a really fastly moving project, which is trying to annotate and discover all cell types in the cortex and ultimately beyond cortex. Do I have a... Right there. Oh, there. This project is part of a cell type program within the Institute, and if you're not familiar with it, I would just like to give you an idea of what it is. The cell types program at the Allen Institute is trying to establish a comprehensive parts list of the brain. And the way we do it is we are trying to characterize these different cells based on genes they express, their shapes, their electrophysiological properties, any property that we can measure in a reasonably fast way. And then what we would like to do is to organize these cells based on, for example, one type of data into so-called unimodal taxonomy, meaning a taxonomy that's informed only by one type of data. Then we would like to also establish correspondence between taxonomies derived based on transcriptomics and electrophysiology, for example. We would also like to investigate conservation of taxonomies across species, and this is what, in particular, we will discuss today in the second part of the talk. We're focusing on mice and humans, if it's not obvious by the shadows. And we would also like to build genetic tools for access to these types in order to really functionally investigate these types and their particular role in specific, let's say, behaviors. We need a way to very specifically perturb them. So we are actually pursuing building genetic tools both for mice and primates. This slide is not supposed to be something to go in details, but to tell you that transcriptomics, one of the particular data types that we will focus in this talk today, has really emerged as a, I would say, initial cell type classification approach. And the reason is the throughput and the ease with which you can define features. If you just assume that your features that you will use for clustering your genes, you can just measure all the genes that are expressed or activated in a particular cell and use those to classify cells. And this is just a summary that's already quite outdated. It's about a year old, where several of us try to kind of summarize the advancements that have been made by single-cell transcriptomics in mice and humans. And what you can see is that multiple different studies have investigated different areas, both in adults and in development. And the number of types that are defined is really staggering, and this is just the beginning. What we have focused on in our studies is the cortex, and this is what we will be talking about today. And I would just like to introduce you to our approaches to transcriptomics. There are multiple different techniques, multiple different both experimental and computational techniques. The way we have approached it is we focus on mice and humans. We are utilizing very similar approach to profiling cells in these two species with some distinctions. So one is that from mice, we actually have an ability to isolate neurons, that is cells themselves from the tissue, and that is very difficult in humans. So one major distinction is that for mice, we use cells, and for humans, we use nuclei. So we do single nucleus transcriptomics. We go after particular areas, and in mice, we will be focusing on two areas: this anterior lateral motor cortex, which is a secondary motor area, and a primary visual cortex. And in humans, MTG, middle temporal gyrus, which is an area that we both can isolate from bank tissue, but also an area that we receive from neurosurgical tissue and that we can pursue additional examinations on this tissue. What we do is we triturate, we protease treat or triturate and triturate the sample to generate single cells. This is for cells, and for nuclei it's slightly different. We sort these individual cells or nuclei, single nucleus or single cell per well. We can also perform manual picking, and especially if the population is really rare. And that is in case when we do, let's say, virus injection to label cells that are that project to a particular area that we want to isolate just based on their projection property. And this is only applied in mice. And then we do relatively standard RNA-seq, but as I mentioned, with single cell per well, to arrive to a particularly deeply sequenced individual cells. So currently on the range of 1 million to, let's say, million for nuclei and 500,000 for cells, but you will see that this first study actually had somewhat deeper sequencing. So in summary, we isolate single cells from defined regions. We analyze the RNA that's in every single cell. We use all the genes, or genes that are differentially expressed higher than the noise, and there are ways to define noise in this system, to cluster these cells, to ask which cells are similar to each other. And I'm just exemplifying these differences in gene expression patterns by different colors. So we use these transcriptome similarities to ask which cells are similar to each other, and we define similarities at multiple different levels. What arises usually is a tree in which nearby groups of cells share many genes that are expressed in them, and as you proceed further down to the root, you arrive to groups of cells that have many, many, up to thousands of genes differentially expressed between them. So I'm going to give you an overview of a recently published data set that recently appeared in Nature. We're focusing here on mice, and we're building taxonomy based on these two regions that I introduced previously, the secondary motor cortex, which is located very anteriorly, and the primary visual cortex located very posteriorly. This particular area is involved in motor planning and motor execution. This one is involved in visual perception. It's the first stop, the main stop in the cortex that thalamus sends information to. We profiled more than 23,000 cells and defined 133 clusters. And what is really, I would say, interesting, striking, is that we define roughly the same number of excitatory types and inhibitory types. Though these come from two different areas, and these come from both areas, these represent roughly 20% of cells, while these represent roughly 80% of neuronal cells. We also define non-neuronal cells, and we didn't go as much in depth with detailing their differences because we have mostly focused on neurons. The most striking conclusion from the paper is that excitatory cells, which are here exemplified by cool colors, are really area-specific. While the inhibitory cells are, in most cases, shared. We have found two types that are primary visual cortex specific so far within this data set. We really find that, so maybe I won't expand much more on this. So excitatory are distinct, inhibitory and non-neuronal as well are shared. We have gone into much more depth than I have time to elaborate here during the talk, but I want to give you one example on, first, whether these fine splits that we define actually mean something. Because, I think each one of these taxonomies should be viewed as a working hypothesis that needs to be examined in other data modalities. And what we need to do is we need to take transcriptomic taxonomy and ask, are there other types of properties that actually match the transcriptomic differences that we see. So what I'm showing here is just focusing on the excitatory portion of the tree. What we see is that excitatory cells follow what we would expect, layer specificity, although fuzzy, not perfect, but there are layer preferences. What I'm showing here is the sampling. So for example, layer 2-3 cells fall here on the tree, and the VISP and the ALM are here together. And then when we look at the sampling, most of them come indeed from layer 2-3. What I will focus on now to address this issue about do these fine differences among cells reflect something about these cells first I would like to say that most of the largest kind of groups of cells follow their projection properties for excitatory cell. So these are intertelencephalic, these are pyramidal tract. I will discuss this particular branch more specifically. And then we have these near projecting and the cortical thalamic cells. So the major branches really follow, major branches transcriptomically follow the projection properties. If we zoom in onto this particular area, ALM, and the projection cells that are subcortically projecting, what we see is that this particular we have two major morphological types that correspond to these transcriptomic type. One type projects to medulla. This is in collaboration with Svoboda lab at Genelia, while the other two types, in fact, project only to thalamus. And this particular dichotomy in projection between these highly related types also correlates with their behavioral roles. The thalamus projecting ones, as this publication has shown, are involved in motor planning, while the other two are involved in motor execution. And with this, I would like to move to where our current studies are on mice, and Zildjian will take over.
So, as the circular just discussed, we'd like to move beyond just two regions of the cortex, and start to build a taxonomy for the whole cortex and the hippocampus areas. So this is an ongoing project, and in this talk I just present our preliminary study on 10 areas that were collected so far, illustrated in this figure. To obtain the highest throughput, we profiled the cells using 10 exonomics, and within these 10 regions, only we collected about 1 million cells, and after QC, about 0.9 million cells are left. So this is a very high quality data set, and here I show the volume plot for the number of genes detected per cell and categorized based on the main cell classes, and we have a median gene detection about 4,000 genes. And clustering these large data sets with close to 1 million cells and tens of thousands of genes presents some technical challenge, and we have now an in-house pipeline to deal with the data sets of this size, but it still has a lot of room for progress. Here, most of our cells were collected on neurons as we fact sorted for neurons. About 15% are GABAergic cells and 80% are glutamatergic cells. And by just throwing this data to the pipeline, we produced 342 clusters. Interestingly, for the GABAergic types, we have 76 clusters. Considering we have about 10 times more cells than what we had in the V1 ALN study, we'll only have a dozen more clusters for the inhibitory types. On the other hand, for glutamatergic types, we have about 5-fold more clusters. So moving beyond, we have 3 main areas to be collected, includes somatosensory areas and auditory areas and olfactory board. Together, we plan to profile 1.3 million cells. Okay. Trying to understand and annotate 342 clusters all at once can be a daunting task. So However, once we organize this into a taxonomy, it becomes more tractable. So the good news is, and the overall structure of the taxonomy is quite similar to what we have observed previously with the V1, and ALN study. And they're broken apart into the inhibitory, excitatory, and non-neural types. And I'll first zoom into the inhibitory types, and we can see that. So boundaries here. Actually, most of the cell types we observed actually are shared across all the cortical areas, and that is consistent with our previous observation. With a few exceptions that some of these types are seems to be specific to the retrohippocampal areas and the hippocampal areas, including entorhinal, subiculum, and hippocampus. On the in excitatory part, we see that most of cell types are regional specific, probably specific to one region or two neighboring regions and things like that. So what perhaps not surprisingly is the hippocampus stands out as the most distinct regions, with some like subiculum cells mixed with the hippocampus cells. So what is also quite interesting, I think, is this the retrohippocampal areas. These warm colors, entorhinal and subiculum, seems to group closer to the cortical areas rather than to hippocampus. So based on some of the conserved markers we have seen previously with V1 AIM study, we can group these excitatory cells into subclasses based, and that's previously seems to associate with a cell with different projection patterns. For example, this whole tree within the orange boxes, based on markers, we think they are corresponding to IT types, which include cells with cortical projection neurons. While this subtree include the PT types, which include cells that project to subcortical areas. Now, with this study, we now think that perhaps all the excitatory neurons, we haven't discussed the layer specificity that basically just mentioned, and they're all present in all these types, that each excitatory types carry a zip code that encoded by this transcriptome that can specify its 3D coordinates within the whole brain pretty accurately. So while we can compute that code pretty easily, the mechanism, organizing principles for this code, and to what extent can we associate the genes that specify the regions and the functions is still pretty elusive. We'd like to zoom into layer 5 PT types, and just because basically just tell you a story about two related PT types within the same region with different projection patterns associated with different behavior. We think this is probably not an isolated instance, but probably a general phenomenon. So, now with this extended taxonomy, we have now identified 50 clusters with total of 24,000 cells in them. And then I showed a two t-SNE plot over here for these the PT types. One is colored by regions, and the other is colored by clusters. You can see the regional specificity is really striking here, and our clusters accurately characterize this regional specificity, and with each region, we have about 4 or 5 clusters. Okay, and here previously the two types in the LM that correspond to different behaviors are here and over here. They don't really look extraordinary in this context. So we hope to systematically set up the correspondence between the transcriptional types and the connectivity, and hopefully some of the will be related to or can be associated with the functions. So so far I have described our efforts with the mouse transcriptome profile projects, and we have parallel efforts to study human cortical cell types, and have many exciting findings. And next, my colleagues, Rebecca and Trygve, will take the stage and share their stories.
Thanks, Suzanne. Okay, so as Basilica mentioned earlier in the beginning of the talk, for human, we've initially focused our efforts on understanding cell type diversity in the middle temporal gyrus of human cortex. And this is because, as she mentioned, this is the predominant area that we receive neurosurgical tissue samples from, and those tissue samples are then used for electrophysiological and morphological analyses that we can also ultimately combine with this transcriptomic taxonomy to create a sort of unified multimodal taxonomy. For our purposes today, so in human, we also have plans to extend our analysis into multiple regions of human cortex, and in fact, we have data from 5 different areas of human cortex now, but for the purposes of today, we'll focus just on this middle temporal gyrus data set. And this data set comprises nearly 16,000 single nuclei. And we used a facts-based strategy to enrich for neurons, so that we sampled 90% new and positive neurons and 10% new and negative non-neuronal cells. And as Basilica mentioned, this is based on a deep sequencing method. So here, for this data set, we used 2.5 million reads per nucleus, which allowed us to detect on the order of 8,000 to 10,000 genes per nucleus. So after using these iterative clustering procedures, we defined 75 different transcriptomic types in human MTG. And here I'm showing a TSNE plot that gives you an overview of all of these different types. And what you can see from this plot is that the types sort of break up into the major groups that you would expect of cell types in cortex. So you see GABAergic interneurons separating from excitatory glutamatergic neurons and non-neuronal cells. And on the right portion here, we've overlaid donor metadata on top of the TSNE clusters. And basically what you can see here is that we used 8 total donors for this study, so 4 of which were post-mortem donors, and 4 of which were neurosurgical donors from MTG. And 2 of the donors were sort of predominant donors, so the cyan color and the red color. Those are 2 post-mortem donors that the majority of the nuclei were derived from. And the point that I want to make with this is that the nuclei from different donors are intermixed with one another within each cluster. So each of these clusters is made up of nuclei that come from multiple genetically heterogeneous human donors. So this tells us that these transcriptomic cell types are relatively conserved across sort of genetically distinct individuals. So in looking at the human taxonomy in a little more detail here, what we see is we define 45 inhibitory types, 24 excitatory types, and 6 non-neuronal types. And there are a few kind of interesting features that jump out from the taxonomy initially. One of which is that the vast majority of the inhibitory types and many of the excitatory types are relatively rare. And by that I mean that they're made up of nuclei, they're made up of a relatively small number of nuclei. So several hundred nuclei or smaller, and that's shown in the bar plot below the dendrogram here. For excitatory types, you can see that that's the case for many of the types, but we do have a few dominant types that are made up of several thousand nuclei. So for example, this cluster here contains more than 2,000 nuclei. Another feature that we noted was that many of the types have a laminar signature. And so because our method makes use of microdissecting specific cortical layers, we can retain some layer of origin information for each nucleus that subsequently gets sequenced. And so we can say where those nuclei are enriched. And the plot here is showing in red layers that have many nuclei that have come from that particular layer. And in white, you have essentially no nuclei have come from that particular layer for a given cluster. And so basically what you can see is that even for the inhibitory types, which we tend to think of as being sort of more sparsely and scattered throughout the cortex, many of them in fact have a clear sort of laminar signature. So for example, these types here in the LAMP5 and VIP branches of the tree, you can see many of them are substantially enriched in layer 1. And the same is true for the excitatory types. They have a sort of broad laminar signature. In some cases, in particular, if you look at this branch, you can see many of these cells come from layer 6. For the non-neuronal cells, that's not so much the case. They generally come from across all of the cortical layers, with the notable exception of this one type that I'll touch on a little bit later in the talk. And lastly, I just wanted to point out that if you noticed from the mouse data, the inhibitory neuron taxonomy in human and mouse looks very reminiscent of one another. So we see major splits into two broad classes that correspond to the sort of putative developmental origins of these inhibitory interneuron types, so the caudal ganglionic eminence and the medial ganglionic eminence. And within these major classes, we see similar division into similar subclasses as well in mouse and human. So looking at the excitatory neuron taxonomy in a little more detail here, there are a few kind of things that popped out to us again that were somewhat surprising. So despite the fact that layers 2 and 3 have undergone significant expansion in human cortex versus, say, mouse cortex, we don't see a huge amount of cellular diversity within these layers. So we see 3 predominant layer 2 and 3 types. We do, however, see a somewhat surprising to us diversity of types sort of centered around the lower part of layer 3 and into layer 4, that we don't necessarily see similar diversity to that in mouse. And the other thing that we noticed as a feature of a lot of the types in human was that, and this is showing again a dot plot where each dot represents a nucleus and tells you what layer it came from in the dissections. And we noticed that many of these types seem to be made up of nuclei that come from multiple layers. And this is a bit in contrast to how we think of excitatory types as being very laminar restricted. And so we wondered if this was perhaps just an artifact of sort of imprecise dissection during the initial collection of nuclei, or if this was in fact a feature of these types in human MTG. And so to look at this in a little more detail, we actually went and did multiplex fluorescent in situ hybridization for 10 different clusters that we defined transcriptomically. And this is now looking at tissue sections where each of these images is a DAPI-stained column of cortex. And the red dots represent cells that are positive for a specific cluster-specific gene panel. And so what you can see is that some of the types are relatively laminar restricted, such as this layer 3C type here. But many of them cross laminar boundaries. And indeed, some of them spread between layer 4 all the way down into layer 6. So they have quite a wide distribution. So this is definitely in contrast to how we sort of think about excitatory neurons generally as being laminar restricted. And it'll be interesting to see if this is a feature of this particular region of cortex, or if this is something that we see in human cortex in general. And so lastly, for my part, I wanted to just tell you briefly about an interesting observation that we had from our non-neuronal cell type taxonomy. And so basically, in human, we find 2 different types of astrocytes, one of which is highly enriched in layer 1, and that's the type that I alluded to earlier, that we see pop out in that previous plot of the laminar distribution of types. And in human and other primates, it's known that this layer of cortex contains a morphologically defined type of astrocyte called interlaminar astrocytes. And these astrocytes have a cell body that sits in layer 1, and they extend these huge processes all the way down into layers 2 and 3 of the cortex. And these are not seen, for example, in mouse cortex. And so what we wanted to do was try and determine if this layer 1 enriched type that we found corresponded to these interlaminar astrocytes. And so we did multiplex fluorescent in situ hybridization in combination with GFAP protein staining, and we were able to show that, in fact, these layer 1 enriched astrocytes do correspond to interlaminar astrocytes, whereas the layer 2 through 6 type of astrocyte corresponds to these protoplasmic astrocytes. And so this brings up an interesting point in that in mouse, in a previous study from Sten Linnerson's group, they also found a very similar distribution of astrocyte types. So they see a layer 1 enriched type. It shares similar high expression of GFAP and aquaporin 4, but it looks entirely different. So it lacks these long processes. It looks more sort of like a traditional astrocyte that you might think of morphologically. But it suggested to us that perhaps there's a sort of core conserved transcriptomic feature of many of these cell types from mouse and human, but differences in a lot of the details of those types. And so we wanted to understand more systematically across our entire data set how mouse and human compared and how conserved the taxonomies were. And so Trygve is going to talk about that now, and I'll hand things over to him.
Yeah, so at a course level, you've seen these 2 taxonomies from human and mouse, and we can see based on even single genes that at the broad class level, these things line up. So there are different subclasses of GABAergic neurons. There are deep and superficial layer glutamatergic neurons, and various non-neuronal types that line up. But really the question remained, how strong, how deep is this homology? Can we line up types at the very leaf level, very individual neuronal types being conserved across 65 million years of evolution? So we have many genes, obviously, to look at. And so we could perhaps initially naively try just a principal components analysis. And if you group mouse cells and human nuclei together, maybe not surprisingly, the first axis of variation is species, and that explains about 20% of the expression variability. So we needed a more powerful technique to align these types, leveraging the fact that many genes share co-expression patterns between species. And so there have actually been a number of these techniques that have been developed for various reasons to look across, to link types across development, to correct for batch effects, and also to compare across species. And this technique adopted this canonical correlation analysis to single cell data and was described in this paper from the Rahul Satij's group. But there are other methods as well, and we've already started trying to validate some of these results with those methods. But here I'll tell you about the CCA results. And just at a sort of broad level, you get much better mixing of the species labeled in these two colors. And on the right, I should say that this is a TSNE projection, a 2D projection of the first 30 canonical correlates. So it's not just 2 dimensions that we're clustering based on. But what we can do is, since these are now overlapping, we can then label the clusters, the 22 clusters that come from this aligned subspace, based on the identities of the cells from the mouse and human data sets. So this is a little bit challenging to see which cell types are actually in each of these clusters. There's another visualization we can use to look at that. I'll try to walk you through this. This is showing human types in rows, mouse cell types in columns. These are just the GABAergic clusters that we've previously described in this talk. And the color indicates the sort of proportion of overlap between those clusters within the 22 that I showed on the previous slide. And so I'll just draw your attention. The easiest to understand really are these one-to-one matches. So these were clusters that included cells and nuclei from just a single type in both species, including these chandelier cells and SST chottle cells, which are this very rare population in deep layers of cortex. And we know about their morphology in mouse, their connectivity, and that allows us to make predictions in human, which is something we can follow up with. But also for just as a side note, when we do this alignment for glutamatergic types, it allows us to make predictions about long-range projections in human, which will be obviously much more difficult to test experimentally. So, and then just the other note is that these data sets are obviously not perfectly matched. We're comparing across different cortical areas, and we're comparing cells and nuclei. So I would really put this as sort of a coarsest resolution scenario. There may be that we're able to match at finer resolution as we get better match sets. But even in this case, so I'm showing here a non-neuronal cell type that we were able to match one-to-one, microglia, and then this chandelier interneuron. And we can ask, how does expression compare between species, even for these very well-matched types? There are a dramatic number of genes that show very large expression differences. So each circle here is a single gene for 15,000 one-to-one orthologs between mouse and human. And 20% of these genes show greater than 10-fold difference, higher in one species or the other. And in yellow, I've highlighted genes that are reasonably specific for microglia. So even very specific cell type markers can shift dramatically between species. For interneurons, glia are actually more dramatically divergent between species, but neurons also have many differences, and about 12% in this particular type. We can also take a gene-centric view. So for a given gene, which types is it expressed in across all cortical types that we've profiled? And so I'm showing, we came up with a metric to sort of measure how much expression is shifted between types, and I'm showing here the top 10 types that came out. I'll just draw your attention to this bottom type here, which shows expression in a subset of VIP-positive interneurons in human, but in mouse is expressed in somatostatin and parvalbumin positive interneurons, as well as an assortment of glutamatergic types. So we can also ask, for different classes of genes, how much has their expression shifted over evolution? And if we rank order these based on the degree of their shift, serotonergic serotonin receptors are the most dramatically divergent. And so, but it's not limited to that. Cadherins differ, glutamate receptors, and a variety of other genes that are very relevant to neuronal function. So I think this is obviously, I think, important to keep in mind for disease models and while people are thinking about the function of circuits. While there's a lot conserved, there are important differences. So today you've heard about these cortical taxonomies that we've defined in mouse and human, but there's a lot of effort at the Institute to now localize these within the circuit, to characterize many different aspects of these types, their morphoelectric properties, their connections, to build tools to label them and perturb them. And then we have now starting up a study to survey variation across human individuals and try to relate that to genetic variation and other variables. And then we have some grant-funded work to extend this to whole brain in mouse and human at high resolution in mouse and an initial coarser survey in human. So thanks to our founder, Paul Allen, and this is really a huge team effort that requires many people's efforts to produce these data sets and analyze them. So thanks to everyone here too. And thanks for your attention.
All right, thank you for that talk. Once again, if you have any questions, please raise your hand and we will bring microphones to you. I think why don't we start over there?
Oh, I don't know what the spatial census is, and maybe it's getting at this question, but my question is about whether or not you've seen any interesting spatial structure in terms of these cell types within areas, right? So within visual cortex or within ALM, are all these cell types, and maybe particularly the pyramidal cell types, sort of uniformly distributed across the cortical surface, or is there substructure within? Obviously, there's substructure within, across lamina, but I'm curious about the other two dimensions across the surface.
Yeah. So we haven't looked at that in depth. So I think we hope to do a survey where so what I was referring to as spatial census is using sort of multiplex in situ methods to label types. Primarily in section, so we can see their laminar distribution, but we can also do in longitudinal sections and see, for example, tiling or neighborhood statistics in that axis as well. I don't know in mouse if they're.
Yeah, I mean, it has been published, but in some cases there is like layer 5, particular like lattice type of organization, and it really would be very interesting to ask that same question with specific cell types. I think we need single molecule fish, multiplex fish, and we need to collect those data because the resolution of what we have now is not sufficient. Yeah.
Here's a question here with Emily.
Hi, I have a question on the hippocampal data. I guess the broad question is how you're thinking about cell type versus cell state when you're making these clusterings. And for the hippocampus in particular, do you see a cluster of newborn neurons, and how much does the transcriptional changes there dictate what cluster they're in versus kind of the eventual cell type that they're going to be?
I think we see some signatures, but it's too early that we haven't really looked that deeply to really annotate all those clusters, what they mean. And we have tons of studies out there, including the one that just published by Mark. So we have to look across all these literatures and try to see who's who and then light them up that way. So, but it's too early to tell that way. But we have tons of clusters and just kind of a little bit overwhelming, yeah.
David. Go ahead.
Hi, excellent talks. Thank you so much for this introduction. I have two questions. One question is, have you been able to identify cell types or that we historically knew are present in the human cortex but not in the mouse, for example, von Economo neurons or the bed cells in layer 5? And the second question, sort of related to that, have you, for the cell types that you think you can find mapping across species and for the cell types that you don't, have you tried to elucidate the potential evolutionary mechanisms for that? So many of us who think about cell types often think about the transcription factors as being the sort of regulators of cell identity. And obviously between mouse and human genomes, there are a number of transcription factors that have emerged over the evolutionary periods, over the 65 million years. So have you tried to look at the enrichment of things like binding sites for those evolutionarily not conserved transcription factors that could have driven the evolution of these cell types?
Okay, well, to your first question, we do have, I didn't describe it here, but we do have data from frontoinsular cortex, looking in layer 5, and had identified a signature of a cell type that matches mouse subcortically projecting PT types. It turns out in human, this is a very rare type, and after doing some digging, we think this is because the subcortical structures have scaled roughly 50-fold greater numbers of neurons in subcortical structures in spinal cord, but 1,000-fold greater in cortex. And so this has had the effect of a sort of 20-fold dilution of these types. So they make up less than 1% of excitatory neurons in layer 5 in human, but about 20% in mouse. And so this is all to get to the point where we thought we had sort of identified a molecular profile for von Economo neurons or for Betz cells, but we're not quite sure. We think that that's actually a cluster that also contains other pyramidal cells in that layer that may not be that very specific type. So, but we're still looking into that, and we're looking for tools that will allow us to enrich for those cells and tease out the PT subcortical projection diversity. To the mechanism point, we have not done any work in that area, but very interested. So I think that's a good way to go, looking at conserved transcription factor expression, for example. And yep.
David.
Well, two questions actually. One is, could you give me a sense? It's beautiful data, by the way. But could you give me a sense as a non-geneticist how much genetic expression is affected by state, like sleep, epilepsy? These samples come from epileptics. You know, the age, the experience of the animal. When you're comparing cross species, I can imagine huge differences in just the history of the human versus the mouse. And
so I could just speak to the human side. So we did, as Rebecca mentioned, we have samples from neurosurgical tissue as well as postmortem samples. And we do see expression differences between those, mostly activity-dependent genes. But those differences are much smaller than the differences between types. So that's why cells sort of end up in their they co-cluster with each other. So, but that's not to say there aren't really important differences, and that's sort of a big piece of the human variation study. But I think in mouse you've actually looked at.
Yeah, so I think when we define these clusters, they can be anything. They can be both types and states. We assume kind of that states will be something that just comes on top of the type, so it may be smaller, but I don't think we have, I wouldn't say we have firm evidence for that. One thing that we do know is this type that we defined in primary visual cortex, a particular somatostatin, a type that we find only in visual cortex. It expresses to a larger degree than many other types activity-dependent genes. So in fact, that particular type may be a state that we only capture in primary visual cortex. So I think for that to really be able to address these questions, we need activity blockers, and we probably need to examine many neurons or many types in different states. We haven't gotten to that yet.
my other question is more of a comment. You only had one functional definition, that was excitatory and inhibitory, and that was the major factor in segregating neurons. And I'm wondering, maybe down the road, when we get really functional information about what these different neuronal groups are doing, it'll become really clean as to what how to separate them.
Yeah, I'm sorry, and I don't think I did such a good job going deeper to the taxonomy, but we have investigated deeper through the taxonomy what has been published on types that we could potentially relate to these types. And we have to use usually marker genes or combinations of marker genes. And I mean, it's very clear that we can find subgroups based on morphologies. So for example, for inhibitory neurons, there is correlation. They also group based on their developmental origins. So medial ganglionic eminence versus caudal. We also find for excitatory types, it's especially prominent, the relationship with projection properties. Are they cortical? Are they subcortically projecting? Are they specifically corticothalamic? We also find this new group that we call near projecting. They don't seem to be projecting anywhere. And excitatory types, you kind of consider they have to project somewhere. And then the layer 6B is kind of a separate group. So even going finer and finer, we see many correspondences with previously described groups. Yeah.
Right, Alex, one more question.
That's a good one. So for these neurons that you see that are of the same type but exist across multiple layers, this might be impossible to do in the human data, but I assume these also appear in mice. Is there any indication that they're actually, do they have different projection patterns? Do they communicate with cells in different layers as well? Or are they essentially interchangeable and just appear slightly anatomically different?
Yes, I would say we don't know. All the types that I showed you, I believe, map to IT types, so cortical types in mouse. But in mouse, I would say, at least based off of the laminar distribution that Basilica showed, we don't quite see the same broadness of the distribution as we see in human. So I think this is still something that we need to investigate a little bit more. But I don't know that we could entirely recapitulate that by looking at mouse.
Great. Let's thank our speakers again.